Hello friends, welcome to Vidya Edu World and our today's topic of discussion is immunity. So friends, in this video we are going to discuss different points like what is the immunity, what are the different types of immunity like innate and acquired immunity, what is humoral immune response, what is cell mediated immunity, what is the difference between active and passive immunity, and what is exactly autoimmunity and allergies. So all these points we are going to discuss in this video. We will start our topic with the definition of immunity. As we all know, the overall ability of the host body to fight against the disease-causing organism by the host immune system is called immunity. So broadly, immunity is divided into two types, innate immunity and acquired immunity. Let's see in detail. Innate immunity is the God-gifted natural mechanism to every living organism. This immunity is also called as natural immunity. This is non-specific type of defense that is present at the time of birth. Okay. As I said, it is non-specific type of defense. That means it is not specific for any particular pathogen, but it can protect us from wide variety of invading foreign agents. So broadly, antigen is the term used for any foreign particles including microbes which enter into the human body. So this immunity provides different barriers to the entry of foreign agents, that means antigens, into our body. So what are those barriers? Here you can see first is physical barrier, which includes skin and mucous coating. Okay, so intact skin prevents the entry of pathogens inside our body, as well as mucous coating of various organs like respiratory tract traps the bacteria in sticky mucus and prevents its entry into the vital organs. Next barrier is physiological barrier. We all know that hydrochloric acid, that means HCl, is present in our stomach for digestion. But it also kills microbes as they cannot tolerate such low pH of the acid. Likewise, saliva in mouth, tears in eyes, they also contain bactericidal component like lysozyme enzyme which kills the bacteria. Next is cellular barrier. This barrier protects us by activating different types of cells in our body, collectively called as immunocytes, against the pathogens. So different cells like leukocytes, that means white blood cells, monocytes, natural killer cells, macrophages, all together create cellular barrier against the pathogen. And last barrier is cytokine barrier. So cytokines are the proteins secreted by the body cells to destroy the pathogens. So different cytokines or chemicals are secreted for the destruction of the pathogen. For example, virus infected cell produced one such protein called interferons, which protect other healthy cells from further viral infection and limits the spreading of virus into nearby healthy body cells. So these four barriers of innate or natural immunity gives our bodies first line of defense. Let's move towards acquired immunity. As the name indicates, it is acquired by host body after encountering with different pathogens throughout their lifespan. Okay, so acquired immunity is acquired by host body after encountering with different pathogens throughout their lifespan. It is not present at the time of birth, but it is earned or acquired by host. Another specialty of this is it is pathogen specific defense. Here you can see it is pathogen specific defense. That means it can identify different pathogens and can produce pathogen specific proteins to kill them. Other interesting thing about acquired immunity is that it is characterized by memory. This means when our body encounters pathogen for the first time, it produces a response called primary response. Okay, so first time response to pathogen is called primary response. And this primary response is of low intensity. We can say this is the first warning to pathogen. But subsequent encountering with the same pathogen gives highly intensified immune response known as secondary or anamnestic response. This is because our body appears to have memory of the first encounter. Okay, so primary response is of low intensity, but the secondary response with the same pathogen is highly intensified because our body has memory of the first encounter. Is it so interesting and mesmerizing? Let's move ahead. Just we have discussed about primary and secondary response. So these responses are carried out with the help of two types of cells, that are B lymphocytes and T lymphocytes. Let's know more about these two pioneers of the immunity. So immune response given by B lymphocyte is also known as humoral immune response. It is a type of acquired immunity. That means it is not present at the time of birth, but it is acquired in the lifespan of the host. This type of immunity is also known as antibody mediated immune response. We all are familiar with the word antibodies. These B lymphocytes produce army of proteins in response to pathogens into our blood to fight with them. So these army of protein are called antibodies or immunoglobulins. So the B lymphocyte produces antibodies against the pathogen. Because these antibodies are found in blood, the response is also called as humoral immune response. As the word humoral is used for relating to the body fluids like blood. Different types of antibodies are produced in our body. For example, IgA. Here Ig, prefix Ig is used for the term immunoglobulin, which is alternate name of antibodies. So types are IgA, that means immunoglobulin A, IgM, IgG, IgE and IgD. So let's take brief information about all these different types of antibodies. So first type or first class of antibody is IgG. These IgGs are the most abundant immunoglobulin, which accounts for about 80% of the total serum antibodies. The key feature of the IgG is that these antibodies can cross the placenta and thus they protect the fetus by giving immunity through these antibodies. Next class is IgA or secretory IgA. Here S stand for secretory, as it can be secreted in body fluids due to its small size and it can be soluble in fluids. It is main immunoglobulin found in mucous membrane in the form of secretory antibodies. So it can be found in mucous membrane of respiratory, intestinal and urogenital tract. The concentration of IgA is found in small quantities in blood, but it is found in high concentration in tears, saliva and sweat. It is the second most abundant antibody and it is found in mother's milk, colostrum. Okay, so the first milk of mother is known as colostrum. So these antibodies also gives passive immunity to newborn babies. IgM is the third most abundant immunoglobulin in serum. It is the largest antibody in size and it is the first antibody to appear in response to initial exposure to antigen. That means after entry of any pathogen or antigen in body, among all other types of antibodies, firstly B lymphocytes will produce IgM antibodies against it. Next class is IgD antibodies. These antibodies do not play direct role in pathogen killing, but these antibodies occurs on the surface of immature B lymphocytes. And last one is immunoglobulin E. It is type of immunoglobulin found only in mammals. And this IgE occurs in allergic responses. So these IgE signals mast cells of our body to release certain chemicals like histamine, which shows allergic reactions in patient's body. Now you know saline features of all the antibodies. You can make chart of specialty of each immunoglobulin for your better understanding. Now here you can see the diagram of structure of an antibody molecule, and this diagram is from 12th standard NCERT biology book. As we know that antibodies are protein molecules, so obviously they are made up of peptide chains, as peptide chains are present in protein molecules. Here you can see Y-shaped structure of antibody molecule. At the tip you can see antigen binding sites. So this region is variable region. Okay, that means for each different type of antigen or pathogen, the shape of this antigen binding site will be different, because to trap or to bind with the antigen, the antibody should be complementary with the antigen, exactly like the lock and key model of the enzymes. So this antigen binding site on antibody is also called as paratopes, which binds to the epitopes on the antigen. Okay, so these are the paratopes which bind to the epitopes which are present on the antigens. So each antibody molecule has 4 peptide chain 1, chain 2, chain 3 and chain 4. Here you can see 2 small are known as light chains and 2 large are known as heavy chains. And all these chains are joined together by disulfide linkages. Okay, so light chain bonded with the heavy chain through disulfide linkage, as well as 2 heavy chains are also linked together with the help of disulfide bond. So this is the overall structure of the antibody molecule. Here you can see some extra points to be remembered. First of all, B cells originate and mature in bone marrow. Therefore, they are called as B cells. Okay, so B for bone marrow. These B lymphocytes divide and form plasma cells and memory cells, which plays vital role against the invading pathogens. As we know, plasma cells actively produce antibodies against pathogens, while memory cells are stored for future immune response against that same pathogen. So these plasma cells gives primary immune response, while memory cells will give highly intensified secondary immune response. Next important point to be remembered is that B cells and T cells can differentiate between self and non-self antigen. So it is very important to distinguish between our own cells antigen and foreign antigens, or else our body can give immunogenic response against our own cells. But our B lymphocytes and T lymphocytes can differentiate between self and non-self antigen, and thus they do not react with our own body's antigen. So this is all basic information regarding B lymphocytes. Now we'll move forward towards our body's another important soldier, T lymphocyte. If you remember, B cells immune response is known as humoral immunity. Likewise, T cells immune response is known as cell mediated immunity, that means CMI. So these T lymphocytes themselves do not produce or secrete antibodies but help B lymphocytes to produce them. The T cells derived its name because they are mainly matured in thymus in the neck but originate in the bone marrow. Okay, so the origin point is bone marrow, but T cells mature in thymus in the neck. Therefore, T for thymus. During maturation in the thymus, T cells divide and differentiate into three broad categories, which are helper T cells, regulatory T cells, and cytotoxic T cells. These T lymphocytes play important role against invading pathogens. Along with T cells are involved in multiple functions like graft rejection. You might know when some human organs like heart, liver, kidneys fail to function satisfactorily. In that case, transplantation of these organs from suitable donor can be a solution. So this procedure is known as grafting. The graft can be any organ or tissues to be transplanted into patient's body. But if donor's graft, for example kidney, is not matching with acceptor's body, then this graft is rejected as patient's body treat that graft as a foreign material. So before grafting, many medical tests are done to check matching between donor and acceptor. So here, point to be noted is that T lymphocytes are involved in graft rejection. Also, T lymphocytes fight against tumor producing cells, that means cancerous cells and virally infected cells along with the pathogens. Likewise, B cells, T cells can produce memory, and these T memory cells have longer memory compared with B cells. T lymphocytes produce different lymphokines which elevate immune response of our body whenever there is a need. So lymphokines are the chemicals or signaling molecules which elevate our body's immune response by activating immunocytes like B cells or macrophages. So up till now we came to know that B cells and T cells are the major black commandos of our immune system. Up to this point, we come to know that what is humoral immunity, what is cell-mediated immunity. But depending upon how antibodies are produced in our body against any antigen, there are two types of immunity, active immunity and passive immunity. If we talk about the active immunity, after exposure of any antigen, antibodies are produced in the host body. This is called active immunity because body actively produces antibodies against antigen. This active immunity is slow and requires some time to give response. If we talk about the examples of active immunity, then injecting the microbes or antigens deliberately during immunization or vaccination is a type of active immunity. Or invasion of pathogen during natural infection is also an active immunity because antibodies are actively produced against antigens in this type. But what happens in passive immunity? Here, ready-made antibodies are directly given to protect the body against foreign agents. Since antibodies are ready-made, here quick responses against antigen is given. For example, colostrum secreted by the mother. Okay, so colostrum is the first milk secreted by the mother which contains antibodies to protect the newborn from some diseases. Another example is antivenom antibodies in case of snake bite. In case of snake bite, immediate antibodies need to be introduced into person's body against snake venom to neutralize the power of venom. In such case, ready-made antivenom antibodies are given to the patients. Hope you get the difference between active and passive immunity. We all are familiar with the word allergy. But what is exactly the condition known as allergy? So friends, the exaggerated or over-response of the immune system to certain environmental antigens is called allergy. And the substance to which such immune response is produced are called allergens. Actually, these environmental allergens are not very harmful, though our body gives over response to it, leading development of allergy symptoms. So in allergic reactions, antibodies produced are of IgE, immunoglobulin E type. Common examples of allergens are mites in dust, pollens, animal dander, etc. And if we talk about the symptoms of allergy, it includes sneezing, watery eyes, running nose, and difficulty in breathing. This allergy symptoms is due to release of chemicals like histamine and serotonin from the mast cells. And in treatment, the use of drugs like antihistamine, adrenaline, and steroids quickly reduce the symptoms of allergy. Okay, I hope you understand the concept of allergy. Let's end up our topic with brief info of autoimmunity. We all know that immunity means body's ability to fight against foreign antigens. But sometimes due to genetic or other unknown reasons, the body attacks self cells. This results in damage to the body, and this condition is called autoimmune disease. Rheumatoid arthritis, which affects many people in our society, is an autoimmune disease. Friends, here we have completed oral immunity topic. Hope you get the topic well. Please like, share and subscribe my channel for more such videos. Confidence and hard work are the best medicine to kill the disease called failure. It will make you a successful person. Have a good day.